Okay, well, thanks very much. It was great to be at this meeting, actually, and I really I'm happy that I was able to be invited to come to this meeting. Our lab actually works primarily on something that's quite different from the main topic of this, direct topic of this meeting, which is really trying to understand the evolution of proteins, protein mechanisms, and really cellular mechanisms. But the model system we use for studying cellular signaling is a photoreceptor neuron from invertebrates. So what I'm going to tell you today is an aspect of our work on that problem, and specifically try to tell you about a certain kind of mechanism that's general in signaling, which is this idea of scaffolding, of having molecules that assemble other proteins and organize them for efficient and local biochemistry. And in studying one such scaffold molecule, which is called INA-D, which is a molecule that operates, as I say, in the Drosophila photoreceptor cell, we discovered something very interesting about it, and about the idea that how scaffolding proteins themselves could be dynamic components that shape the signaling process, but actually gives us an idea of how these things could even evolve. So this is what I would like to go in the end of my talk today. All right, so what's the model system? All of you probably seen this picture. The invertebrate eye is, in the case of Drosophila here, is a really beautiful structure. It's a crystalline array of unit eyes, each of which are called ommatidia. And I'll just remind you that the ommatidium contains a number of support cells and pigment cells and so on, but also includes 8 photoreceptor neurons that are the only light-sensitive cells of the eye. And the purpose, of course, of the ommatidium is to convert light energy, photon flux, into a graded electrical potential that forms the input to downstream processing centers in the brain. Now, about 25 years of work has really gone into defining the molecular mechanisms by which this so-called visual transduction process operates, and I basically can summarize it here. Not all the players are here, but the main players are here. The membrane, the light-sensitive membrane, is full of the light receptor, which is a member of the G-protein coupled receptor superfamily you just heard, so this is rhodopsin. When rhodopsin absorbs a photon of light, it initiates a cascade of signaling events, so works through, in this case, a GQ isoform of heterotrimeric G protein, which then activates an effector molecule, which in this case is phospholipase C beta, which through its usual catalytic mechanism, which is not important right now, ultimately opens a non-selective cation channel of the family of trip channels. Trip channels, as I say, are non-selective cation channels, and you get then, when they open, an influx of sodium and calcium ions, and that represents the electrical response of the cell. So a response to a photon of light is a transient opening of these channels and a brief depolarization of the cell membrane. So if you do experiments where you electrically record from these cells and you give flashes of light that were so weak that on average only a single photon is being absorbed by the cell, which means one receptor molecule is activated, you get these elementary signaling responses that are called quantum bumps. Basically, there's a Poisson distributed or randomly distributed wait time followed by an all or nothing electrical response that represents the opening not of one channel but more like 30 channels, which then very rapidly shut, and then you get a brief refractory period before a next event can happen. The macroscopic response to light with the simultaneous absorption of, say, 10,000 photons is nothing more than a statistical superposition of all these single photon responses. So basically, if you understand the dynamics of this thing, you understand the essence of this, the visual signaling process. Now, a key aspect of the shaping the response is feedback regulation. Turned out that the calcium that flows in the cell also initiates its own secondary signaling activity, in this case via turning on a protein kinase C, which then phosphorylates some components, including the channel, and causes the response to decay rapidly, so the response can be very brief in time and you get high temporal frequency responsive vision. Now, one of the interesting players in here that I've not mentioned is the pink molecule, which is a scaffolding protein. It's a concatemer of just 5 bits of molecular Velcro known as Pdz domains. Each Pdz domain binds to, we think, one of the components of signaling. In fact, here are the assignments. So the 5th Pdz domain is thought to bind the effector molecule. The 3rd one binds the ion channel itself, and the 2nd one we think binds this protein kinase C. And the point is the scaffolding molecule assembles all these proteins into a single macromolecular complex, which is kind of the essence of activation and feedback regulation. We were very interested in understanding the role of the scaffolding in shaping the visual response, particularly because there were genetic studies that showed this was really important. So if you knock out the scaffold, all the components are there in the membrane, but the flies are essentially blind. The thing is the total knockout of vision. If you make point mutants, like a mutation in this Pdz domain that knocks off the interaction with the effector molecule, that effector molecule is there in the visual membrane and so on, but the failure to make this association makes the flies what I call legally blind, right? They make quantal responses, but they're disorganized in time, and they're slow and small. As a result, you never get a superposition of these responses to give you a significant macroscopic response. The effect is the flies are basically insensitive to light. And other mutations give you other features. So basically it was interesting to understand how this organization at the scale contributes to a physiologically critical property in this fast vision. So we started solving actually structures of pieces of vena D. And one of the first things that came was just the structure of just the fifth Pdz domain of vena D. And you would think ordinarily just solving one component of a domain of this family would not be such a big deal, but actually we learned an enormous amount from the structure. Let me show you what that structure showed us. So to start off with, what is the Pdz domain and what's the canonical structure of Pdz domains? So here's a structure that was reported many years ago by Doyle et al., the rat brain sort of first Pdz domain that was solved, and it's really the prototype for the whole family. These are, as I say, bits of molecular Velcro. They bind other target proteins. So here the target protein, which is bound, a little short peptide sequence of the target is shown here in green. We would describe this protein basically as a 6-stranded beta sandwich with a couple of asymmetric helices, and there's a groove formed on the surface here between that beta strand and this alpha helix, and the ligands bind there. So this is sort of the so-called active site or binding pocket of the Pdz domain. What was interesting was we crystallized and made very nice crystals of the fifth Pdz domain of vena D. So now this is just the fifth Pdz domain taken in isolation. The structure was solved by traditional methods in X-ray crystallography, and I'll happy to answer methodological questions if you wish. But the idea was with atomic resolution, when we looked at the protein, we saw that there were similarities but some significant differences from canonical Pdz domains. In particular, here's a comparison to show you what I mean. Here's an alignment of 4 distant sequence distant Pdz domains. The first 3 are other eukaryotic domains, Pdz domains. And here's an overlay of the structures of those first 3 sequences. And basically, despite enormous sequence divergence, they all have exactly the same structure. You can see that here. The fifth Pdz domain of vena D is different in 2 ways. This backside alpha helix is rotated by roughly 60-65 degrees. But more interestingly, the active site, the binding pocket, is completely distorted in this domain. The helix is essentially unwound. There's a couple of tight turns of 310 helix, but the active site is not in a configuration that could bind ligands in the traditional way anyway. The reason for this distortion was also obvious in the structure, and that was that if you look in the electron density, in the core of the protein, the middle of this protein, there's an intramolecular disulfide bond, a cysteine that emerges from this helix and one from the third actually beta strand, form this clear disulfide interaction. And clearly this is the simplest hypothesis for the distortion here. Now this actually caused us great, I would say, worry at the beginning, because the most likely explanation for this is utter nonsense, right? Because the disulfide bonds generally do not form in the inside of cells, because the inside of cells is generally maintained in a rather reducing environment. And this seemed to us as there was a potential this could simply be an artifact of the way that the crystallization was done in vitro. But there were a few things that made us actually very interested in the idea as maybe physiologically relevant. The first was that the bond occurs in solution and that it's an extremely strong disulfide bond. And I'll show you that data in a moment, but so strong that it could easily persist in the inside of nearly any eukaryotic cell. So to show you how we figured out the standard redox potential of this disulfide bond, we basically use a chemical technique known as maleiamide reactivity. The basic idea is that these chemical compounds like maleimides will react with free sulfhydryl groups on proteins. So when the cysteines are engaged in a disulfide bond, there's no reaction that's going to happen. But as you incubate the protein in stronger and stronger reducing conditions, you can get a reaction of the protein to these maleimides, which causes a mobility shift in a SDS page gel. So you can see the protein being converted from an oxidized species to a reduced species over a range of external reduction potential. And that basically allows you to work out what is the standard redox potential, effectively, of this disulfide bond. And the answer was that it was nearly minus 0.4 volts. Which, what does that mean? The inside of most cells is about minus 180 to 250 millivolts. So this suggests that this bond can not only form, but it's really a challenge to explain how it could even ever be broken in the inside of cells, actually. So it seemed like it was potentially relevant. There's one other argument that I will skip for the interest of time. But this basically allowed us to proceed with this project. And what we did was re-derived crystals under different conditions, where we grew crystals now under conditions of 10 millimole dithiothriitol, a reducing agent. So at this concentration of DTT, there should be an equilibrium between reduced and oxidized species of this protein. And when we solved the structure here, and again this was done by now conventional X-ray diffraction methods, we found a new structure actually. So there were, in this case, 3 molecules of this Pdz domain in what's called the asymmetric unit of the crystal. That's the minimum repeating unit of the crystal. So inside the asymmetric unit, the 3 molecules are free to adopt different conformations. And in fact, what we found was all 3 molecules were adopting different configurations in one crystal, actually, right? So here's what we got. The blue molecule was exactly the same as the original structure. Disulfide bond was intact and the structure is distorted. But the green molecule, now the disulfide bond is broken, right? So now you have no disulfide bond, and the structure you see has snapped back to that of this traditional structure of all other Pdz domains, where the binding pocket is now open. The red molecule is just like the green. The disulfide bond was broken and the active site was open, but by chance the C-terminus of the neighboring molecule was placed in its active site, so sort of pseudo-liganded, if you will. We don't believe this is the natural ligand for this domain, but it is demonstrates that this configuration is capable of binding ligands in the same manner as other Pdz domains. So basically, in one crystal structure, we got 3 things, right? We got the closed, the oxidized state, the reduced apo state, and if you will, a reduced pseudo-liganded state, which I thought was quite lucky of us. But the result is, in the end, it tells you there's a possibility that this scaffolding domain is actually able to adopt 2 configurations, one in which the active site's open, one in which the active site is shut. And the idea is that there's a possibility this could be actually a physiologically important transition in the molecule. And so, of course, this makes you ask the question, this sort of 2-position switching of this domain, is it biologically relevant? Does it actually happen in vivo, first of all? And is it regulated by light in the photoreceptor cell of flies? So how do you do this kind of experiment? So first thing we did was we created some reagents. We did the obvious experiment, which is to mutate one of the cysteines, actually this one there, to serine, which is an amino acid where you could not get the disulfide bond. So in principle, in this mutant, the protein should be locked in the open or reduced configuration. We did a crystal structure of the cysteine to serine mutant, and in fact, we found in the structure only the configuration of the reduced state, and in fact, the protein looked exactly the same. Even the rotamer of the cysteine was converted to serine, but nothing is different except for that the sulfur atom now is turned into an oxygen atom. That's basically, this is very clear that this mutant is a good mimic of of locking the molecule in the reduced state. And then we made transgenic flies in which we put back a mutant form of INA-D that contained this point mutation, now transformed into knockouts where don't have any INA-D. So functionally, we're replacing the endogenous INA-D with a form that is locked in the reduced state. And here's the expression of the transgenic flies to show you that we're expressing normal levels of this now mutated protein. So what's the effect? How do we see this disulfide bond forming in vivo? Non-trivial experiment. And Sean Mishra, the student in this work in the lab, did an incredible job in developing this assay. So here's how it goes. What you do is you take flies that are grown either in the light or the dark, and you cut their heads off in the dark, and then you directly homogenize the heads into 25% trichloroacetic acid, which basically precipitates all the proteins in the head. So this is a way of freezing, if you will, the state of all of the molecules in their disulfide bond state without any further reactions happening, because you immediately denature all proteins here. And then you resolubilize the proteins, everything in the head, under denaturing conditions. So what do you have in your tube? You've got proteins that many of which have free sulfhydryls. I mean, every protein in your head has probably at least a cysteine in it, so there's a lot of free sulfhydryls, but then some of them may be disulfide bonded, but they're denatured proteins. And then Sean developed a 2-step maleimide reaction assay to specifically look at disulfide bonds. The idea is simple. You first react with a non-fluorescent small maleimide, which effectively blocks, if you do it right, all of the free sulfhydryls that are in all the proteins in the head. And then you come in and you use a reducing agent to reduce any disulfide bonds that were there, creating now new sulfhydryls, which then you can react with the fluorescent maleimide and in principle label only disulfide bonded proteins. So let me show you, and then you run these in a 2D gel and look. So let me show you how the assay works. So if you take the denatured resolubilized proteins from the head and directly react them with the fluorescent maleimide, you get what I told you. Every protein in the head is lighting up, so you get a big mess on the gel. But now if you block with AMS, forget to reduce, and then try to react with the fluorescent maleimide, nothing is labeled because you've effectively blocked all the sulfhydryls with the AMS reagent. But now if you do the experiment correctly, where you block with the AMS and then reduce, and then treat with the fluorescent maleimide, you see a small number of proteins labeled in the 2D gel, which are now the disulfide bonded proteins in the head. And in fact, one of them was clearly the case of was INA-D. So here's the interesting experiment. If you raise flies in the dark and you do this experiment, you don't see any spots appearing here. That means there's no disulfide bond, free sulfhydryl, sorry, there's no disulfide bonds in this protein that's labeled here in the dark. But if you take those flies and you give them a flash of light and you activate the visual system, and then you look, you see the appearance of these spots. So this is light-dependent formation of a disulfide bond in some protein in the head. The protein is INA-D because if you then take immunoblots and look carefully at the position at these spots, in fact, you can show that these spots are the INA-D scaffolding protein. If you treat flies with light and then you let them relax in the dark before you cut their heads off for about 10 minutes or so, then this disulfide bond relaxes. So this is the evidence of a reversible formation of a disulfide bond pumped up with light and then relaxes back as the fly's dark adapt. So transient formation of a disulfide bond in the INA-D protein, and controls. So if you knock out INA-D, you don't get these spots obviously in the light. But more interestingly, if you take our point mutant that's locked in the reduced state, so no disulfide bond can form in the point mutant, you also do not see these spots in the light. And what that means is the disulfide bond here that's forming in vivo in a light-dependent manner is the disulfide bond which we are looking at in our crystal structures of the 5th Pdz domain of INA-D. So that was a beautiful evidence that in fact this disulfide bond switching of this scaffolding domain is happening in vivo. All right, what's the physiological meaning of this light-dependent sort of conformational switching of the scaffold? So if you look at quantal responses, I showed you these earlier. Wild type have this property of having this short, randomly distributed wait time and a rapid quantal response to light. Point mutants in INA-D show you various pathological effects on the quantum bump, but the point mutants that are locked in the reduced state have absolutely no defect at this quantum bump level, and that's an important control. It says that by locking this protein in the reduced state and by making transgenic flies and so on, we didn't really do anything. We fully rescued effectively vision at the low light levels. But what was interesting was that when we started doing experiments where you challenge the photoreceptor cells with larger amounts of light, more physiological levels of light, we started to see a pathological response in the mutants that are locked in the reduced state. So what's shown here is there's a black trace, which is wild type flies. There's a blue trace, which is a transgenic animal in which we have put back a wild type copy of INA-D into the INA-D knockout mutant, and you can see that fully rescues the wild type like response. The red one though is the mutant where it's locked in the reduced state, and you can see as you increase the light intensity, there's a progressive decrease in the rate at which the visual response is inactivating. So there's actually a 3 or 4-fold defect in the relaxation of the visual response when the INA-D scaffolding molecule can't switch into this oxidized state. That's interesting. It's a cellular phenotype for this thing, but it makes me want to ask the question that does the fly care about this? I mean, it's interesting that there's a 3-fold decrease in the visual response, and certainly that's statistically completely non-random. There's no way the wild-type flies ever show this effect, but does a fly care about this? What's the effect on the animal? And so to look at this, what Sean did was we set up a behavioral assay, which is a specific one to look at fast timescale visual signaling. And here's how it goes. There's a natural, evolutionarily conserved behavioral response in the fly, which is common to all flies, actually, called the escape jump response. And the idea of this response is that if a predator comes over their visual space, you get a shadow over the eye, and what the flies see the shadow and execute canonical behavior where they jump backwards and take off flying. So it makes flies very hard to catch, actually, right? And so this escape jump response is mediated by a system in the fly where the visual input or even mechanosensory input is integrated into a neuron called the giant fiber neuron that innervates muscles in the leg and thorax that produce the behavior. You can simulate the escape jump response by giving flies a visual startle. That is, to take flies and you give in the normal light and you give them a brief period of darkness. And if they have a proper escape jump response, they'll jump in response to a brief period of darkness. Okay, and of course the question is, by decreasing the frequency, temporal frequency of the visual response in these mutant flies, have we disrupted this high frequency visual function phenotype? Let me show you the results of this. First of all, if we look at a very low frequency assay for visual response, which is just phototaxis, which is, say, can flies just see the light and walk to the light, you find that wild type flies are in black. They do it 60 some odd percentage of the time. If you look at the mutants that in which we put back wild type INAD into the INAD knockouts in blue, you get a full rescue of the phototaxis phenotype. But also you get full rescue of phototaxis in our point mutants that are defective in this conformational switching. That makes sense. This is a low frequency visual response, and visual function is basically normal in these guys at the low light levels anyway. But in contrast, a knockout of rhodopsin shows you a completely knockout phenotype and even phototaxis. But now if you look at visually mediated jump response, you find that while putting back wild type INAD can rescue you just to the same level as wild type flies in their behavior, these mutants that have a threefold defect in the kinetics of their visual response effectively a complete knockout in this evolutionarily conserved escape behavior. So this is a functionally critical feature of this conformational switching in the INAD molecule for important behavior. Of course, flies that have no rhodopsin can't do it either. But important control, it's nothing like we disrupted about the visual about the startle response itself, because if you do a mechanically mediated input into the same giant fiber system, everything has a proper escape response. So this shows you that, and this is just a movie to show you. Let me show you this really quickly. Oops, sorry. So here's wild type flies. If you give them, you'll see in a second, a brief period of darkness will happen, and the fly basically dematerializes where it shows up somewhere else. They're very fast, right, these guys? So this is a normal escape jump response. But if you now look at these mutant flies that are knocked out in this conformational switch in the scaffolding protein, when you see a brief period of darkness in here, he could care less. He just doesn't see the darkness at all. Okay, so I want to say one last thing before I'm done. What is the mechanistic basis for this? For getting this inactivation defect? I've shown you the inactivation defect is behaviorally critical, but what is actually going on in the mechanism? So I'll just give you the bottom line, because it's not time to tell you the answer. You can read about it here if you wish. But the bottom line is that I told you already, the properties of the quantum bump are a randomly distributed wait time, the shape of the response, and something called the refractory period, like an action potential in central neurons. There's a period of refraction after the creation of a quantal response. It turns out, I'll just tell you the bottom line, that this inability to undergo this conformational switch in response to light is a complete knockout of the refractory period following a quantum bump. So these animals have the capacity to have secondary events in this period where there should be no visual signaling, and that gives them the degradation of their frequency response of vision, and consequently the behavioral responses I showed you. So to summarize what I've told you, this little story, it turns out that you know, the scaffolding proteins were thought to be passive, non sort of actively participating components of signaling, which are just there to put things together in space and localize. In fact, what this shows you is that INAD PDZ5 occurs in two states, one which is normal and one is distorted, and that light actually triggers a transient switching of the scaffold into an oxidized configuration and then returns upon dark adaptation, and that mutants in which we locked it in this configuration have these severe defects in both inactivation and visual behavior. The bottom line in the end, and the idea that this is involved in the refractory period. So the model in the end is that INAD, when it's in this configuration, is signaling competent, but upon transient oxidation to this configuration, it produces a refractory period, which makes the membrane unable to signal further for a brief period until the molecule returns to the state. And this switching then is critical, this refractory period, for getting high frequency visual responses out of this system. So the summary is we conclude that INAD is actually more like a dynamic machine, I suppose, rather than a passive scaffold, undergoing conformational changes on basically the time scale of visual signaling, hundreds of milliseconds or less, to shape the visual response. And I want to leave you with one final idea. There's a lot of questions in principle one could follow up on this project, which I think we probably will do some of it, but not much. For example, one could ask the question, how does light actually trigger this switching? If you want to ask me in the question period, I'll tell you the first order answer to that. There's a question of how, once you switch to this configuration, you become refractory mechanistically. And if you want to ask me after the question session, I'll give you the first order answer to how that works. But these to me are not actually the interesting questions. The interesting question is, how does this kind of a system evolve? I mean, you've got here a fancy allosteric conformational switch in a Pdz domain, which has not been observed in any other Pdz domain. And yet, if you look in the evolution of flying insects, you find that this system is only conserved in the daytime living flies that need fast vision. If you look at the midges and mosquitoes and the nocturnal flying insects, they don't conserve this conformational switch mechanism. So it's really interesting for me to pose the question of how some sort of a molecular evolution at the level of atoms in amino acids and proteins are controlling a behavioral response, which is presumably controlling reproductive fitness of the organism in its natural condition. Okay, so with that, let me stop. All of this work was effectively done by one terrific graduate student, Sean Mishra, who's now a postdoc at Caltech, working with David Chen. And I want to thank the people that paid the bills. Thank you very much.
Thank you, Rama. It was fascinating. Specific question to this presentation? Okay.
That's very nice. I was just curious, in the different forms of your crystal structures, are there any changes on the back side of the molecule?
Well, no. We don't, in response to this conformational change, if you really look carefully, I don't think you can conclude that there's really any other conformational changes except for the local ones you see in the active site in this case. It's not to say such changes may not be there, particularly in dynamics and so on, right? But there's no crystallographic evidence that that's the case in this instance.
A second quick question. Have you tried engineering a second disulfide bridge sort of further up the helix? So that, you know, further up the sort of canonical binding cleft. In other words, to sort of kind of mimic the rigidity that you're enforcing by the one that you've
So why would we do this experiment? You want to see whether we could reproduce the same kind of distortion, you mean?
Not to reproduce the distortion. I mean, when I first looked at it, I thought, the disulfide just effectively makes the molecule rigid. So I wondered if you could engineer a disulfide at a different place further up the helix, across, across the cleft that would mimic that.
It's an interesting experiment to do, but I'll tell you another piece of data. We've done a more comprehensive analysis of mutational analysis in Pdz domains, and it turns out that the stability of the domain has really very little correlation to its function. And so the idea that there's rigidity and stability in this protein is somehow going to be deeply tied to its function, I don't see any evidence for that in the data. But it's an interesting experiment to try.